I have a question here for you, and this question is interesting, it's important, and this is conceptual. So, see the question here. The bond between the phosphate and the hydroxyl group of sugar is a or an what? Do you think it should be glycosidic bond, or it should be ester bond, or it should be phospho and hydride bond, or it should be peptide bond? Well, to answer this question, what we need to do is talk about chemistry. So this question will have a good part and a bad part. For some of you, it can be bad because I am going to talk about a bit of chemistry over here and structures. But the good part here is if you just understand that, these type of questions will be really easy for you to answer. So, but I would say you did not worry because I am here. I will make this easy for you. So very easy structure. See, you know about DNA, you know about nucleotides. What comprises a nucleotide? There is a nitrogenous base, there is a ribose sugar, there is phosphate, right? So if we know that, so we should be able to draw it. So this is the structure. I'm drawing the ribose sugar. So here I have the base. So I'm just writing as base, which is attached over here. Fine. Now here what I'll have is H, I'll have OH, here I'll have HOH, fine. Now here what I'll have is H and CH2OH. This is the typical structure. Now I have not added the phosphate, okay. Now another thing you should know is the carbon numbering. The numbering starts from here, 1, 2, 3, 4, 5. So this is the 5th carbon. Okay. Now, I told you about the phosphate. So, phosphate PO43 minus, I represent it, would look like the structure is this. Fine. Now, this structure I've drawn is a deprotonated structure. If I draw a protonated structure, how will it look? I can write it as this. Just add OH, OH. It's a protonated structure. Now, how the reaction is going to happen, how the phosphate is going to attach itself to the ribose sugar and the base which is already attached. So base is attached to carbon 1, and now what happens, this OH you are seeing, okay, this OH will be attacked over here. Now let me say, this is a very crude way of saying this. The exact way would be this. How will it get attached? Attachment will be P double bond O minus by O and O minus. This is the attachment that will happen. So now we have a nucleotide, and this bond that has occurred over here is called ester bond. You should remember this is the ester bond. Fine. Now, don't get confused. What is an ester bond? There's a basic criteria for something to be called as an ester bond. Let's imagine there's a carboxylic acid. Carboxylic acid, you know, very basic structure. C, we have C double bond O, yep, OH. Perfect. Now, carboxylic acid, along with that, we need to have OH of the alcohol group. So R is the alkyl group, you know. So this is represented it as R for the carboxylic acid and R prime for the alcohol. Now here is the reaction that will happen. So OH from the carboxylic acid and H from the alcohol will be removed. That means water molecule, one more water molecule will be removed, and what I'll get is this. I'll have C double bond O, I'll have R. Now I'll have the bond which is this. Okay, perfect. Yes, this is the ester bond you're seeing over here. This is the ester bond. So you understand, H2 has been removed, OH from carboxylic acid, I said you, and H from what? H from the alcohol. Fine. So we are left with O. So this bonding is called the ester bond. Just see this criteria. Now we'll go back and see the same criteria over here. See, there is P double bond O It's just not for carbon. Okay. You see here P, this is pentavalent. And carbon, it is tetravalent. Here, pentavalent, I have P double bond O. And I had OH over here, double bond O over here. So, this is almost similar to the carboxylic acid I have shown you here. See, it had double bond O, OH. Correct? Yes. So, OH from phosphate will be removed. And I had over here what? I had CH2OH. So, H from this is an alkyl group. It is attached to the ribose. So, this is the alkyl group of, like I represented alcohol over here. Okay, you understand the similarity? Yes. Then it's easy for you. So, H will be removed from this OH of CH2OH, and OH from the phosphate group will be removed. And the bond that will happen via O is this, and this is called the ester bond. Understand what is the ester bond? Now, it doesn't end here. There's a particular terminology used often for this is phosphodiester bond. Now, the question is, why it is a diester bond? Diester means there will be two ester bonds. We have discussed about one ester bond. Where is the second one? Okay, fine. Let's do the second one also. So for that, what I'll need is another molecule of nucleotide. Again, H, we have the base over here. Of course, we have OH, H, we have OH, H, and yes, we have CH2, and will be linked with the phosphate, correct? O minus, O minus. Fine. Now, we have the diester. Now, where do you think this will happen? Again, numbering 1, 2, 3. This is the third carbon, fourth and fifth carbon. Perfect. So now, what will happen? See over here. So, let me draw the protonated form for this again. This is OH. I told you the protonated form will be OH and OH. Correct? Yes. Now, what will happen again? This, as you remember, this is quite similar to that I talked about. In an esterification reaction, we need C double bond O. Okay. So, this particular reactant, this is quite similar to the phosphate group over here. We have P double bond O and we have the OH group over here. See this molecule. See this ribose over here. It has OH. So H from this particular third carbon will be removed. So what do we have? Yes, this OH will be removed. This H will be removed. So by O, there will be a bond formation. So I'm just tilting it and drawing. So rather, in fact, I could have represented it as like this. So here I'll have O. So I'm just showing the proper bond formation. Okay. I'll have what? Double bond over here. Yes. So what do you think? This is what type of bond? It is again an ester bond by the phosphate. So now, do you understand why it is called phosphodiester bond? Yes. There are two ester bonds that is happening over here. Fine. Phosphodiester bond. So now I think it is clear for you. The answer would be what? It's ester bond. See, the bond between the phosphate and the hydroxyl group. Hydroxyl is OH. Phosphate, I told you. Okay. Phosphate and the hydroxyl group of sugar is A or an what? It's an ester bond. Option B is the right answer. So we'll of course discuss the other options also, because these can help us in answering other questions related to these options. Fine. Glycosidic bond, very simple, between glucose molecules. Okay. Quickly I'll show you. So this is a glucose molecules and it will be linked by this. Okay. This is a glycosidic bond. Fine. This is the glycosidic bond. Simple. Two glucose molecules I have shown. Here, of course, I have CH2OH. Similarly, here also I have CH2OH. So this is the glycosidic bond. Simple. Linking two sugars. Now, what about this phosphoanhydride bond? Have you heard about this? Let me tell you, you have already encountered in this question about this bond, but we have not discussed it. Now, it's time to discuss. See this. We always talk about ATP, adenosine triphosphate. Adenosine triphosphate. here I have drawn one phosphate. So this is not the correct structure. You know why? Initially, these nucleotides that we have are in the form of triphosphate. So the proper structure should be this. The proper structure would be here. Okay. It will again be linked to another phosphate. And yes, we have two. It will be linked to another O. The phosphate, double bond O. Here. This is the structure. Okay. You see triphosphate 1, 2, 3. Correct? Triphosphate we have. Fine. And just to ensure that whenever you draw structures, just check the bonds. What I told? Phosphorus. Okay. It is pentavalent. See, here it is pentavalent, satisfying 5 bonds. Here also it has 5 of 1, 2, 3, 4, 5. Okay. This phosphate also 1, 2, 3, 4 and 5. Okay. Pentavalent. So our structure is correct. Fine. Now this is a triphosphate. Triphosphate. Okay. Now this triphosphate is really high in energy. There are 2 high energy bonds. Okay. So these 3 phosphates are joined by the 2 high energy bonds. And what are they? The high energy bonds are, let me write it here. This bond particularly and this bond. These are the 2 high energy bonds, and just recall when we talk about any reaction where ATP is used, that is adenosine triphosphate is used, then after using we always write ATP converted to ADP plus what? PI. So what is ADP? Okay. So adenosine diphosphate, every time these 2 phosphates are released one by one, then this results in the cleavage of this bonds. When this bond is cleaved, it is high in energy. This energy is released. Okay. So yes, these are the bonds which links these triphosphates are called what? They're called phosphoanhydride bond. Perfect. I hope you will not forget this. So this is not an answer, but you now know what is a phosphoanhydride bond. Peptide bond, it's very simple. Peptide bonds are what? They join the amino acids. So yes, the amino acids, very simple. We have O, we have OH, and we have N, H, H. So the more we keep on drawing structures, and this is one of the way I personally use so that I can keep remembering the structures. Okay, so keep drawing the structures. It will be really easy for you to remember the structures. H. And what will happen? This H and OH will be left. So we'll have H2 which is coming out, and the bond will be C and N bond. Okay, this is a what? Peptide bond. You'll find it in polypeptides, proteins. So well, this question was really interesting, and I am sure right now you understand how the DNA nucleotides are formed. The DNA is formed. What are the different types of bonds over here? What is this phosphoanhydride bond? What are the ester bonds that is happening? Why they are called ester bonds? Why they are called phosphodiester bond? And let me tell you, you'll get a lot of questions related to this terminology, phosphodiester bond. Now I'm sure you can answer any question related to these. So it's an easy diagram. Just remember it. And yes, logically you can answer all questions based on these structures. So answer for this question is option B, that is ester bond.